And then let's see if I can do a laser pointer. Okay, awesome. Okay, so hi everyone. My name is Gizem. I'm a research assistant in Michael Levin's group at Tufts University. And today I wanted to tell you a little bit about my take on this exciting field of Biobots. This is my first EBICS meeting ever. So this could be like maybe a little too high level, but yeah, we'll see how it goes. And I specifically want to emphasize how going forward in this field, I think that this concept of self-construction can really help push the field forward. So I'm going to start with this sort of my kind of map of the Biobot landscape. So here I wanted to plot the current examples and their capacity for self-construction, sort of on the y-axis against like what they're made of. So at one end of this y-axis, we have this sort of kind of lower capacity for self-construction, where a lot of top-down form-giving equipment is used, such as like 3D printers, molds, or more advanced UV lithography techniques. And sort of on the other end of the spectrum, we have the embryo, this kind of bottom-up morphogenetic capacity for developing form. So for the other axis, the x-axis, I put the sort of constituents, like what they're made of. So we have abiotic examples where we see this like purely chemical and mechanical driven shape-shifting behavior using a diverse set of systems like shape memory alloys, pneumatics, or electroactive polymers. But none of these biobots are necessarily made of living agents. Then we have these hybrid systems where hydrogels often are seeded with living cells, pretty frequently muscle tissue. Or we actually, pretty recently have this entirely biotic systems where both the structural integrity and the function of these bots are actualized by living cells only. So finally, the sort of question I'm asking here is, now that we have bots that are made entirely of living cells, which we know have the capacity to build entirely on their own, an incredibly diverse set of forms and functions that we see in nature, can the next biobot jump to this level where it can self-construct, perhaps from single cell? So before sort of discussing further how that might look like and why that's necessarily better, I wanted to zoom a little bit more into the existing examples. So that I've kind of clumped into different types here. So I'm not necessarily going to elaborate too much on type 0 because since it doesn't necessarily include any living organisms, which are the only sort of programmable building blocks that we have right now that's capable of self-replication and self-construction, I'm kind of going to gloss over it. But having said this, there are some really exciting examples live there. For instance, there's this work by Jennifer Lewis's group. I mean, this is just one example. There's so many like this, but they printed these composite hydrogel architectures that shapeshift. They swell and shrink thanks to the anisotropic properties that they have. So it's kind of like wood, actually. And again, there are many other examples I encourage you to kind of dive into if that's something you're interested in. So next, I'm really going to kind of focus on these type 1 biobots, which again is a intricate sort of constellation of polymers and cells. So they are really the way I see them, they're really the pioneering examples of biobots, and sort of this mascot is one of the early examples is the Metasoid. I assume that everybody's familiar with this, but I'm still going to quickly explain. So here they are using rat cardiomyocytes, seeding them on a silicon polymer, and they are stimulating them with in an electrical field. and causing this sort of biobot to propulse itself. And then a work, except that this time we again see this, engineered muscle tissue, but this time the stimulation is made optogenetically. Again, when we shine light onto this, this time I believe this is skeletal muscle. When we shine the light on the skeletal muscle, we also see it shape-shifting. And then yet another example here, it's constitutive, so there isn't necessarily induction going, but the kind of like as a like different approach from the previous examples. Instead of cardiomyocytes, they use skeletal muscle. Yeah, I love this video. So it's kind of self-propulsing and again moving forward. So, and I know that there has been so many new versions of these, this like multiple legs, etc. But I want to put these as the early examples. And just to kind of like summarize and put this back into that plot that I had, here we see hybrid composition and we see different modes of actuation. So here there is a special sort of equipment requirement in that you would need like 3D printers or some like both for making them and also for deploying them to actuate them. You would need like optogenetic induction infrastructure. So because of this all like specialized equipment, I feel like the throughput is maybe not as high as we see in some other self-constructing structures in nature. So one would make like one at a time. So which kind of place, so this kind of explains why this example is placed in here. So the a similar construction, so just kind of going forward, a similar construction logic where the bots are built kind of top down, the form is imparted top down, is the xenobots. I guess like one advantage of this way of building, both with type 1 biobots and type 2 biobots, is that because we're building top down, we have a really like high resolution control over where the cells go, and we can really we have control over cell count. We have control over which type of tissue interacts with what other type of tissue. So it definitely has a lot of advantages. And the biggest difference here, looking at type 2, is that here we have these systems composed entirely of cells. So for this, as far as I know, systems that are self-propulsing and that is composed entirely of cells, Xenobot is the only example that I'm aware. If I'm overlooking anything, please let me know. So I'm quickly going to, since this is rather new literature, I'm quickly going, this is not my project. It is from our lab, so I'm going to be explaining to the best I can, but if you have specific questions, I can definitely put you in contact with my colleague who worked on this. So here the design process, the workflow starts with a computer sort of running this evolutionary algorithm and using two different types of building blocks. One of them is sort of this more like static ectoderm tissue, which is shown as red here. And the other one is, again, this like, they're cardiac cells from frog. So they're also able to propulse themselves constitutively, sort of contract constitutively. So what is being done here, once the computer sort of develops these different forms, optimal shapes are selected based on a predetermined fitness threshold. And then this embryonic stem cells from frog is being carved into these specific shapes. And it's not only the shape that kind of corresponds there, also the tissue type. So the red ones here is the self-contractile cardiac tissue lined with this more static ectoderm tissue. And when it's sort of compared head to head with the simulation, where the the blue one, actually, I'm going to put the negative control too. So the blue one is the in vivo behavior, the actual organism behavior, and the pink one is the in silico. We see that it matches very closely, and actually there are certain cases where the in vivo behavior even exceeds the expectations of the simulation. So again, sort of, and I actually do have a video too, just to kind of show. Okay.
Coming to the end of the time we have, so maybe just
okay.
more points. Yeah, just to give people a chance to ask their questions.
Yeah, definitely. Okay, thanks for the reminder. So again, this just kind of like summarizing. The main thing, I guess, here is that even though this is entirely made of cells, because it's not sort of self-constructing, again, a sort of skilled like scientist needs to carve them. So throughput is still quite low. And so for the kind of just coming to the last class, the type 3, which I guess is going to be informed together by the community. So I don't have specific examples for this yet. But just to kind of give an idea of how this could happen, we can certainly imagine having a genetic circuit put into a single cell, and then that cell self-replicates and build this cluster of cells that interacts. And I know that Dr. Weiss thinks a lot about this. But one thing sort of I wanted to also add here that we can do is maybe we can think of putting these DNA circuits not only to sort of blank cells like CHO cells, but maybe instead we can think of putting these into more like stem-like or progenitor cells that are already good at creating certain functions on their own, either in matrices, when grown in matrices. And this way we can kind of relieve the pressure from the circuit a little. And I think that that could be one way in which we can think of doing self-constructing bots. And so because they are, so here I just want to kind of circle back into the couple questions that were asked. So this could help us make these device-free biobots that can be deployed outside the lab or device. I mean, we can imagine, of course, doing these inducible programmables so that they can go into the body and do useful work. And because no like special skill set or equipment would be required, as they're self-constructing, we can say that like they're autonomous. And most importantly, we can make a lot of them in parallel. And that could help us sort of go translational because we can mass produce and mass test them. Yeah, and also we can imagine sort of if we can make like hundreds of these, we can imagine kind of getting inspired by nature. We can imagine these bots interacting with one another and as a group creating these even larger shapes or even structures, because we know that these swarms in nature not only create these shapes but also kind of connect to one another and form these load-bearing structures like ant bridges. So I'm just kind of thinking if we can have this self-construction framework, we can create such biobot swarms and make them create larger and larger structures. Yeah, so I'm going to stop here. And if you have any questions, again, I don't know if this was like too high level, but if you have any high points of sort of conversation, I'm happy to address.
We have about time for, I'm going to say one question, just okay. on track.
So Gizem, that was great. This is Ron. So would a type 4 be biobots that can build other biobots? And would there be advantages if it's not things that are just self-constructing, but ones that can construct other biobots? Is there something that's not perhaps possible just with self-construction?
So is your question like, can these bots
make other bots?
Make other bots, yeah. I mean, they're not mutually exclusive, I guess. You can make self-constructing bots that can also then construct other bots as well. But you can, so like as one emergent behavior, we know that bots let alone like in a field, can aggregate things together. So if you need to kind of build your bots not out of a single cell, but out of the aggregation of multiple cells, you can imagine bots aggregating individual cells into, bots like themselves. So I guess even there, like, but even there, you need kind of like a swarm-like behavior. I don't think that like a single bot can make a second bot, but maybe like 10 bots can make like 3 bots, sort of harnessing that collective behavior there.